Hi everyone, it's Richard again from RedShiftBio. And today I'm going to give you a presentation on an application note that's available on our website entitled Microfluidic Modulation Spectroscopy as a Non-Destructive Structural Characterization Technique, a case study using bovine serum albumin. First, and as usual, I'll give a brief introduction to our instrument and technology. The picture shown here is our second generation of instrument, the Apollo. It's powered by the core technology of RedShiftBio called Microfluidic Modulation Spectroscopy, or MMS. These tools are used for characterizing protein structures at a previously unachievable sensitivity and reproducibility. You can check out the link down below for more information about our products. So what is MMS? MMS is an advanced IR technology. Instead of using two different cells, MMS utilizes a single microfluidic flow cell that can modulate between the sample in solution and the referencing buffer. This is essentially an auto-referencing mechanism that can happen at every second, and ultimately, the average differential absorbance is recorded across the AMITE1 band. This rapid autoreferencing mechanism can largely minimize cell movement during the measurements. And as a result, it drastically improves spectral reproducibility. MMS can achieve over 98% system repeatability for just 2 mg per ml protein samples, and it gets higher as the sample concentration goes higher. This means that it can detect smaller than 2% of structural differences between samples. Finally, because of the design of the microfluidic flow cell, MMS is a non-destructive technique that allows us to recover samples after analysis to a recovery rate of at least 95%. In this app note, we'll use BSA as a test case to demonstrate the non-destructive feature of MMS. To design an experiment where we can do a head-to-head comparison of the samples before and after the MMS analysis, we first produce a 5 mg per ml BSA sample. We used PBS as the buffer in this case to make it more of a realistic sample. We then ran this sample through the system and obtained the first set of results. Now, again, because MMS is a flow system that simultaneously injects both the sample and the reference buffer for auto-referencing, these two eventually mixed into a single line. So we ended up collecting a diluted BSA sample that is roughly half of the original concentration. So next, we divided this collected sample into 2 portions. The first portion was immediately analyzed by MMS to obtain the second set of results. And by using a protein concentrator, the other portion was concentrated back to the pre-analyzed volume with the goal of matching the original BSA concentration. Finally, this concentrated sample was analyzed by MMS to generate a third set of results. These 3 sets of results after each analysis were then compared. Now let's take a look at the results. First, here showing the differential absorbance spectra of the 3 samples across the AMITE1 band. We can see that the 3 samples are all similar in shape, but different in intensities of this 1656 peak. This peak is a signature peak for alpha helix, which is the predominant secondary structure motif of BSA. One thing to mention is that each sample was measured in at least 3 replicates, and the spectrum for each replicate is actually shown on this graph. But as you can see, they are visually indistinguishable for each sample. This essentially demonstrates the extremely high sensitivity and repeatability of MMS measurements, which is particularly important when you're trying to determine very, very small differences in structure between samples. And now because of this, MMS is also a great tool for quantitation. If we plot the peak intensities of each sample against the calculated concentration, we can see an extremely strong linear response with an R square equal to 0.99999. As you can see in this table, the calculated concentration of the original BSA sample was actually 4.75 mg per mil. The reconcentrated sample, on the other hand, is 4.5 mg per mil, which is roughly 95% of the original sample. And therefore, we can say that the recovery rate in this case is 95%. In order to compare the similarity of the samples, first we converted the differential absorbance spectra to what we call absolute spectra, which are essentially normalized by concentration. Now, if I draw your attention to these red plots for the replicates of the original sample, we can once again see that they are highly overlaid, just as expected. And next, this is the collected sample in green. And finally, the reconcentrated sample in blue. As you can see here, all the spectra of these three samples are still visually indistinguishable, just like the replicates within each. But how do we determine the similarity of these samples in a more scientific approach other than visually the same? To extract smaller spectral details, we can convert these absolute spectra to the second derivative plots. And this time, the spectra are not completely overlaid, and you can now see some tiny spectral variations. And to get the similarity scores between these samples, we can inverse the second derivative plot and baseline it to get a similarity plot. We can now use the area of overlap of these plots to get two metrics: repeatability, which is the similarity between replicates, and sample-to-sample similarity using the original sample as the control. And these numbers are listed in this table. As we can see in the second column, all the three samples maintained over 99% of repeatability, meaning that the system variability of these samples are just under 1%. With that being established, now if we look at the third column, the similarity scores of the collected and reconcentrated samples in comparison to the original sample, we can see that they are both over 99% as well. This indicates that there's indeed no structural change of this BSA sample before and after the initial analysis, as well as after the reconcentration procedure. If we take a step further using the higher order structure analysis tool of our software, we can obtain the relative abundance of each of these four secondary structural motifs of BSA. So it's well known that BSA is an alpha helix rich protein, as you can see in the crystal structure. This is in agreement with our analysis here that's showing roughly 80% of alpha helix structure. In addition, the HOS analysis once again shows that there's no structural difference between these three samples, as indicated by the same relative abundance of each secondary structural motif. In summary, we showed that MMS can be used as a non-destructive protein characterization technique with extremely high sensitivity and reproducibility. We use BSA in PBS as our test sample and show that the structure of BSA maintained over 99% structural similarity before and after MMS analysis. In addition, samples run after MMS can be recovered using a protein concentrator, and the recovery rate using this method can reach over 95%. The structure of BSA sample after reconcentration also maintained 99% compared to the original sample. This abno has demonstrated the ability to recover, reuse, and reanalyze samples after MMS measurements, which is particularly beneficial for studies where samples are extremely precious, ultimately saving time and cost having to produce more materials for future experiments. Thank you so much for your attention, and I hope you find this video useful. If you want to read the full app note, you can find it on our website under the Resources tab. Take care, everyone, and I'll see you next time.